Hey everybody, welcome back to General Biochemistry Lecture. We are doing the second part of Chapter 27, which is on protein metabolism. The first part we left off with just getting the amino acyl tRNAs ready for translation. Now we're going to pick up with still the initiation process, but we're going to be taking the ribosome and acquainting it with the mRNA and some tRNAs, and we're going to get the whole process moving. So let's continue with protein synthesis. So the first stage, like I said, getting the tRNAs ready, getting them charged with the correct amino acid, and that process in itself contributes to the fidelity of the protein synthesis process, making sure that you have the correct amino acid charged to the correct tRNA. The second stage, we're still in initiation. We have to make sure that there's a specific amino acid that actually initiates protein synthesis, and that's the AUG initiation codon. All organisms have two tRNAs for methionine. There's one for when that AUG is the 5' AUG, that's the initiation codon. And then there's the one when you have an internal methionine residue within a polypeptide. In bacteria, there's a difference in terms of the methionine is actually, it's modified. So the beginning methionine has a formal group at the amino terminal end. So this is actually added in response to the AUG initiation codon. The MET tRNA that's charged with methionine inserts methionines into the interior position, and those are added in response to the interior AUG codons. So there's a difference for bacteria. The initial 5' AUG that starts initiation, and then the interior ones. So there are two different tRNAs. There's FMET tRNA, which is the formal and formal methionine, and that will insert at the amino terminal end. It's the very first amino acid. So this modified amino acid is formed in two successive reactions. So the first one is summarized by saying that we're going to charge the tRNA that's specific for the formulated methionine. We're going to charge that with regular old methionine. Then we're going to take that methionine charged tRNA, and we're going to transfer a formal group from tetrahydrofolate to the methionine. THF is my abbreviation for tetrahydrofolate. So that's what happens. You can charge that tRNA for FMET with just plain old methionine. And if it does not get methylated or formulated, rather, then you can actually use that in the interior too. But it needs to have that formal group added onto the methionine residue in order for it to initiate translation. Now that's bacteria. Eukaryotes don't add a formal group, although in mitochondria and in chloroplasts you will see that. But we have to differentiate between what happens in the cytosol, and then what happens in the mitochondria of vertebrates, and what happens in the chloroplasts in plants. The mitochondria and chloroplasts are almost like their own entities within the eukaryotic cell. So in eukaryotic cells, we have a specialized initiating tRNA that inserts methionine at the N-terminal end. And we only have the one AUG codon for methionine. So it's the same one that also inserts, we have to also insert the methionine into the interior positions. So we've got the same codon, but we've got a specialized tRNA for starting with AUG and something else for inserting the interior positions. So we're going to be going back and forth in this one between bacterial and eukaryotic stages of translation, really doing kind of a comparison. Again, in general, the difference between bacterial and eukaryotic processes is that the bacterial ones are usually simpler, fewer proteins, just less complex overall. Eukaryotic processes tend to have more proteins, more regulation steps, that kind of deal. But if you boil it down, they're very similar procedures. So initiation in bacteria requires quite a bit. And if you remember, I showed a table that had all these different factors that are necessary in the different stages. Now we're going through all of those things bit by bit. So in bacteria, we first need the 30S ribosomal subunit, the mRNA, that initiating tRNA that's charged with the formal methionine. We need some initiation factors, GTP, magnesium, and we also are going to need the 50S ribosomal subunit. Remember, the ribosome is in two big pieces, one big and one small, though they're both very big. And they have to come together and kind of form a sandwich with the mRNA going through that cleft between the two. So we first have to form an initiation complex. This should sound strikingly familiar to DNA and RNA metabolism. In this initiation complex, we've got the 30S subunit, and we've got two initiation factors, IF3 and IF1. They prevent the 50S subunit from adding on to the 30S subunit and combining prematurely. We have to load the RNA, the messenger RNA first, and we have to get that mRNA aligned so that we have the start codon lined up where we need it to be. And part of how that happens is the Shine-Delgarno sequence. And this is a region of the mRNA that guides the initiating AUG to its correct position. And the 16S ribosomal RNA, which is a part of the 30S, there's some complementarity there, which helps to identify that Shine-Delgarno sequence, and that will position the AUG in the right spot. So we're going to talk about the E, the P, and the A sites in a second. So I know you see the E, P, and the A, and it may jog your memory a little bit, but don't worry, we're going to talk about them in a second. I first want to talk about the Shine-Delgarno sequence first. So you're going to notice that there's lots of A's and G's, and that's because it's a purine-rich area that comes right before the starting AUG. So in general, it's going to be somewhere in the ballpark of like 8 to 10 base pairs, or I should say nucleotides. There's base pairing between the Shine-Delgarno sequence and the 16S rRNA that is within the 30S subunit of the ribosome. And so there is some base pairing that occurs that you're going to see help position that messenger RNA in the right spot. So there are two places that you're absolutely going to see some kind of loose binding, and that's here on the ends. But then you can see this region here and we're looking at a consensus sequence versus the 16S rRNA. You're going to see some real good base pairing here and lots of hydrogen bonding, and that's going to stabilize the mRNA in that position so that the AUG is properly positioned and you get the right frame to read the correct protein. Because remember, this is all about the correct reading frame. If we're one off, then we're not going to get the right start. So bacterial ribosomes have 3 sites that bind tRNAs. Eukaryotes do too, so this applies to both systems. There's the aminoacyl site, or the A site, and that's where the incoming aminoacyl tRNA comes, aside from the initial methionine. The initial methionine binding is a different beast, but all the other amino acids, when they come in, it's the incoming aminoacyl tRNA. So it's charged with the next amino acid that's going to bind. IF1 binds here and prevents the initial tRNA from binding at that site during initiation, because we don't want it there. We actually want that AUG codon lined up with the P site, the peptidyl site. And that's where amino acids are added to the growing chain. So the P site is where you're adding on to it. This is the only site where the initial methionine tRNA can bind. The E site, or the exit site, binds the uncharged tRNAs, and that's kind of the last stop before they dissociate and leave the whole ribosomal complex. So let's get back to forming that initiation complex. We've got the anticodon of the F-met tRNA that's charged, and it's correctly paired with the initiation codon at the P site. So IF3 and IF1 are blocking the E site and the A site respectively. The only place for that tRNA to bind initially is the P site. To facilitate this, there's another initiation factor, IF2, that binds GTP, and it will bind the 30S subunit and recruit that special methionine tRNA to base pair with the start codon. Once we have this complex, then we recruit the 50S subunit to create a complete initiation complex. GTP is hydrolyzed in the IF2, and all of the initiation factors leave. So you can see how already we've used a lot of energy. We've used energy to charge all of our tRNAs with amino acids. We've used energy to recruit the starting methionine to the correct location. And now you're going to see how much energy we use just to simply add on amino acids to a growing chain. So we've got the initiation complex, which is the 70S complex, which is comprised of the 50S subunit, the 30S subunit. We've got the mRNA bound in the correct position with the AUG, the starting AUG codon, in the P site, and we also have our special methionine tRNA bound as well. This is our initiation complex. In eukaryotes, the eukaryotic mRNAs are bound to the ribosome as a complex with specific binding proteins. So like I said, it's always a little bit extra with the eukaryotic cells. Eukaryotic cells have at least 12 initiation factors, and just by the mere saying of at least, it means that we still don't know. There are at least this many, and this is functional, but there could very well be more. We have some functional homologs of IF1 and IF3, and so they are, the E is for eukaryotic, IF initiation factor 1, so IF1 is in bacteria, EIF1 is in eukaryotes, and there's EIF1A, B, etc. Then we also have a similar thing with IF3 in bacteria and EIF3 in eukaryotes. So they have a similar function, and we'll see that in a second. There are some other players that we're going to talk about that create the initiation complex for eukaryotes. One of the major differences in this whole protein synthesis process is that in eukaryotes, we actually circularize the mRNA. In bacteria, we don't. And you'll see later on that the processes of transcription and translation in bacteria are actually coupled, whereas they are not coupled in eukaryotes. So there's another initiation factor, EIF4G, that binds to the poly-A binding protein at the 3' end of the mRNA. So remember the structure of a messenger RNA, you have the 5' cap, which is shown in red here. And then there is also a poly-A tail at the end, the 3' end. And there's proteins, poly-A binding proteins, that bind that part. And they help to, 1, protect the tail from being chewed up. And 2, they also help interact with the cap and circularize the messenger RNA. And they do so through interactions with EIF4G. There's also, I want to point out, an untranslated region, or a UTR. And there's a lot of information there too that tells the machinery kind of what to do. So that untranslated region, even though it doesn't end up in the protein, it definitely has purpose. So all of this is assembled on the 40S subunit, and there's the similarity here of the smaller subunit from the ribosome is where the messenger RNA will bind first, and then the larger part is recruited later. So in eukaryotes, we have additional factors that get added along with the mRNA to make a 48S complex. And this complex will scan the mRNA until the AUG is encountered. So there is no Shine-Delgarno sequence here. It's just this massive complex scanning the messenger RNA until it finds the starting AUG. This could be facilitated by an RNA helicase that's part of EIF4A. So we've got this major complex. We've circularized our mRNA. We found the starting methionine. And now we have to also bind that tRNA with methionine. Because remember, the E site and the A site are blocked, just like in bacterial protein synthesis. So EIF1 and EIF3 are there. They help to make sure that the mRNA binds, that the tRNA does not bind before its time. There's also 2 different GTP-bound initiation factors, EIF2 and EIF5B, and we're going to talk about those next and how they help recruit things. So the 60S subunit associates with the complex once the AUG has been found. EIF5 promotes EIF2 GTPase activity. So the GTPase activity just means hydrolyzing GTP. And once that happens, that EIF2 that has GTP bound, when it hydrolyzes GTP, it has less affinity for the initiator tRNA that it helped recruit. So it's going to fall off of the complex. EIF5B also hydrolyzes GTP, and it helps to trigger that dissociation of IF2. So it's kind of both and. You have this GTPase activity that stimulates the release of all of these initiation factors. What you're left with is a ribosome that is fully assembled. You have an initiation complex with the methionine charged tRNA bound to the start codon. You've also got some proteins that are bound to the 5' cap that are helping protect it. And you've also got things at the 3' end that are helping to protect it and to circularize the whole thing. So a little bit more complex, but in terms of the timing and telling all of the initiation factors to release, it's still based on GTP hydrolysis. And that timing of the hydrolysis is what helps to time the whole process of assembling the ribosome. So here are the protein factors that are required for initiation of translation. We've got bacterial here at the top, and all these eukaryotic ones. So there are lots of factors, and they serve some of them overlapping roles. So this is going to be quite a bit to, keep straight in your mind. But these summary tables will be very helpful for you when you're studying, when you're doing quizzes and things like that. So make sure that I usually don't promote like printing things out and stuff, but having this handy would make your life a lot easier when you're trying to study, or you have a few minutes, maybe you're on the bus, or you got a few minutes, you're waiting for something to happen. You can just kind of whip it out and look at it. So now that we've got our initiation complexes formed, we can now go ahead with elongation. So we're going to go back to our bacterial example and talk about what's required there, and then we'll bring it to eukaryotes. So elongation requires the initiation complex that we just made, all those amino acyl tRNAs that we made in the pre-steps to get ready. And we need elongation factors, which in bacteria we need EFTU, TS, and G. The EF stands for elongation factor. We also need a ton of GTP. So what has to happen with our initiation complex is we have to have the binding of an incoming amino acyl tRNA, and then that tRNA has to transfer its amino acid to the initial methionine, and a peptide bond needs to be formed. So how does that happen? There's an incoming amino acyl tRNA that binds to EFTU, and EFTU has GTP bound to it. That whole complex will bind the A site of the 70S initiation complex. Remember that the A site is the amino acyl site where you're going to add on to, it's going to provide the new amino acid that's going to get added onto the chain. EFTU will hydrolyze its GTP, and it will leave the ribosome. And during the time of that hydrolysis, it gives the ribosome a little bit of time to figure out, okay, is this a right fit or is it not? The next thing that we have to do is form a peptide bond. So the methionine that is there, the starting methionine, is transferred to the amino group of the amino acyl tRNA in the A site to form a dipeptidyl tRNA. That just means that you've got a tRNA So let's draw these sites. We've got the E site, the P site, and the A site. The E site is empty right now. What we do have is my sad attempt at a clover leaf. We've got one tRNA bound that has MET. We have another tRNA bound that has some other amino acid. We'll just call it AA for amino acid. So what happens is you form a bond between these two, a peptide bond, and then that transfers this methionine over to this tRNA that's in the A site. So what you're left with, and I'll draw it down here, is you've got an empty tRNA in the P site, and then you've got a tRNA in the A site that now has the starting methionine attached to the next amino acid via a peptide bond. Once that happens, you have to shift the empty tRNA to the E site, and the tRNA with the two peptides, our growing polypeptide chain, has to shift to the P site. And that leaves the A site open for a new tRNA that's charged to bind. So let's look at the formation of the first peptide bond. We can't do this without looking at the chemistry. If you thought we were going to get by without doing that, wrong. Got to look at the chemistry. So we've got a tRNA with our F-MET in the P site. We've got our new amino acid. Don't care what the identity is, because aside from proline, adding on amino acids looks the same. We've got the lone pair on the nitrogen of the amino acid in the A site that's going to attack the carbonyl carbon of the carboxyl group on the methionine. We're going to form a tetrahedral intermediate, because the electrons in that double bond in that carbonyl, that's going to go up to the oxygen. And then the oxygen is going to say, you know, having all this extra charge is great, but I really liked that double bond. I feel like that was more stable. So we're going to come back down and reform that double bond. And the connection here with that 3' hydroxyl, well, those electrons are going to go out and grab the hydrogen off of the 2' hydroxyl. And you no longer have attachment to the tRNA in the P site. You now have a bond between the F-methionine and that second amino acid. To complete the reaction, this 2' hydroxyl is like, okay, so now I don't have a hydrogen. I'm feeling kind of lonely and cold. So I'm actually going to steal a hydrogen from this amine over here. And that's how you form this amide bond. So those electrons go up to the nitrogen, and we have our amide bond between these two amino acids. Once that happens, we now have to shift, and the ribosome is just going to shift, and we're going to kick this empty tRNA to the E site and this newly elongated polypeptide attached to this tRNA to the P site, so that when the next amino acid that's charged to a tRNA binds to the A site, it will do the same thing. So you should be able to draw this. So you should be able to draw how this happens and how you elongate a polypeptide chain. The exception would be proline. So if you have to add 2 prolines in a row, then it gets a little bit weird. The ribosome doesn't like that. Proline doesn't have a free amine. It's a secondary amine. Proline just causes all types of trouble. So speaking of prolines and causing trouble, we've got to talk about ribosome pausing, arrest, and rescue. Ribosomes will sometimes stall, and reasons for that include damage to the mRNA, or if it's an incomplete mRNA. Remember, RNAs are very tasty for nucleases. There are enzymes that are just chomping at the bit, waiting to chew up that messenger RNA as soon as it is exported to the cytoplasm. That's why we have the 5' cap, we've got the poly A tail. All of these things are protective so that if a nuclease does get a little bit excited and starts chewing away, that there's some protection. The 5' cap, a little bit harder for a 5' to 3' exonuclease to chew up. Likewise, the poly A tail gives it something to chew on. All those A's are not going to make it into the protein, so you're not chewing up the actual message. But sometimes those nucleases win, and they damage some RNA. What happens is, and again, we're still in bacteria, we have tmRNA that rescues stalled bacterial ribosomes. This transfer message, or tmRNA, pretty much comes in and gives the ribosome another substrate so it can keep going versus just being stalled in that one spot. So what happens is you have an mRNA that's got a stalled ribosome on it. Say it doesn't have a 3' and stop codon. This transfer messenger RNA, or tmRNA, has a 5' end that looks like a tRNA. And so it will bind in the A site along with EFTU, hydrolyzed GTP, and it adds an alanine to the peptide chain. Now we have transferred the polypeptide to a new RNA. And we can now translocate this tmRNA, kick off this empty tRNA, and add on new tRNAs just like we normally would in translation. And we can continue on until we get to the stop codon in this transfer messenger RNA. That stop codon will allow the ribosome to terminate and the ribosome to be recycled like it normally would. And this protein that's made, the sequence of it gets tagged for degradation by proteases in the cell. And we're actually going to talk about protein degradation at the end, because that is part of protein metabolism. So the process makes a protein that is tagged for degradation so that it doesn't go out there and, mess up some other stuff, interacting with something it's not supposed to interact with, what have you. But this is a way to rescue those ribosomes and allow them to keep working and being productive versus just getting stuck forever and waiting for something else to happen. So there's the 23S ribosomal RNA that has peptidyltransferase activity. And this is the ribozyme part of the ribosome. It catalyzes the reaction by binding and aligning the tRNAs in the A site and the P site in their proper orientation. And if you think about a protein, all the enzyme active site is a place where the chemicals, i.e. the substrate and the amino acids, are positioned in such a way that the chemistry will happen more readily. And that's what this RNA does. It positions the tRNAs so that the chemistry of making that peptide bond will happen spontaneously, quickly, and without a whole lot of energy input that you would have to do on the benchtop. There's a very highly conserved active site A residue. And when we say A, we're not talking about alanine. We're talking about A as in the nucleotide in the 23S rRNA that may facilitate this reaction by either general base catalysis and transition state stabilization, or potentially some other method. But this is still, it's accepted, but there are still some question marks and still some possibilities for other potential methods for this reaction to occur. But this A residue is very highly conserved, and it clearly has a role in this process. So we talked about how we form the peptide bond. We talked about how ribosomes can get stalled sometimes, and they need to be rescued. But if that doesn't happen, or even if it does, you still have to translocate, which means the ribosome has to move forward one codon towards the 3' end of the messenger RNA. So this is going to shift all of those tRNAs one site over. So what was in the P site will now be shifted to the E site. What was in the A site will be shifted to the P site. The A site is now open for a new aminoacyl tRNA. The translocation of the ribosome requires elongation factor G, or EFG, and the energy that is provided by GTP hydrolysis. So if you'll notice the trend that we have these elongation factors that are GTPases, and the hydrolysis of GTP is coupled with some kind of big event, whether it is the binding of the tRNA that's charged in the A site, or it is translocating the whole ribosome. GTP hydrolysis is a timer, and it provides energy for the actual chemistry and the moving of parts. So in bacteria, what happens is you have GTP hydrolysis of EFG. And EFG is actually in the A site. So after we have that initial F-met that has the amino acid transferred to it, and then we do the translocation, EFG binds to that A site with a GTP bound. It hydrolyzes GTP, and after that, it has GDP bound, and it leaves. And that makes the A site available for a new tRNA. So just the hydrolysis of GTP provides the energy for that to happen. The structure of EFG actually mimics the structure of the EFTU in complex with tRNA. So if you look at these two structures side by side, this is EFG on the left with GDP bound, and on the right we have EFTU with a tRNA bound. they look pretty similar. So this allows EFG to bind the A site and displace the peptidyl tRNA so that it can then move to the A site, or excuse me, to the P site. After this, it's just wash, rinse, repeat. The elongation cycle keeps repeating. You attach a third amino acid residue in the same way that you did the second. For each amino acid residue that's added, you're hydrolyzing 2 GTPs. So let's go back to part 1, where we said that those long open reading frames have somewhere in the ballpark of 500 plus codons, which will roughly translate to 500 amino acids. And we're using 2 GTPs for every one that we're adding. So you can see how this is a very, very, very high cost to make a protein. And we definitely don't want to have to do it more than we have to, which is why protein synthesis is so heavily regulated. So let's talk about the eukaryotic elongation cycle, because that was just the bacteria. Now I've got to bring it forward to eukaryotes, talk about how much more complicated they are. But honestly, it's pretty similar. So there are elongation factors in eukaryotes that have analogous functions to the bacterial ones. So EF1 alpha, EF1 beta gamma, EF2. And those are all analogous with EFTU, EFTS, and EFG. Notice how, again, there's the lowercase e for eukaryotic. When a new amino acyl tRNA binds to the A site, there's an allosteric interaction that leads to the ejection of that uncharged tRNA from the E site. Now we have to talk about proofreading again. We talked about proofreading in a sense when we talked about how the tRNAs are charged with amino acyl tRNA synthetases, and that there's a secondary site that will accommodate improper amino acids. So in the case of the isoleucine tRNA synthetase, valine is very, very similar. But the very small difference between valine and isoleucine, that one methylene group, is the difference between valine AMP fitting in that secondary site and being hydrolyzed, and isoleucine going on to being added to the tRNA. So that was the first proofreading example we had. Now we've got proofreading on the ribosome, and I hinted at this a little earlier. The GTPase activity of EFTU contributes to the rate and fidelity of protein synthesis. So in that pause time, where the EFTU GTP is bound to the tRNA, and in the time that we have that hydrolysis occur, and then EFTU GDP leaves the complex, that exists for a little bit. So it takes a few milliseconds for all of that to happen and dissociate. So if there's an incorrect amino acyl tRNA in that site, there's time for it to dissociate from the A site. Remember that if you have a tRNA that does not, so it has the anticodon that's supposed to hydrogen bond with the codon on the mRNA. If there's a mismatch there and it actually doesn't bond as well, then there's time for it to say, oh, you know what, I don't think this is the right fit for me, I'm going to leave. So that GTP hydrolysis that EFTU has, not only does it provide energy, but it provides timing. It provides the little bit of time that allows the ribosome to not just automatically incorporate a tRNA right away, but it gives time for those hydrogen bonds to actually settle in and say, okay, yep, this is the right thing, or for that tRNA to leave. There's a GTP analog, GTP gamma S, that has one of the oxygens on the gamma phosphate replaced with a sulfur. And it is used all over the place when you want to slow down GTP hydrolysis. And when you use that with EFTU, it will slow hydrolysis and it will improve fidelity because it gives longer times for the ribosome to proofread and say, oh, you know what, this doesn't match. But this will absolutely reduce the rate of protein synthesis because you're pausing for longer times. And it can actually also stall the ribosome on the messenger RNA. And it can, reduce the rate of protein synthesis because you're stalling the ribosome out. And that's not good. But it's a great way to study enzymes that hydrolyze GTP. You can kind of give yourself a longer window to look at what's going on. So we have elongated our polypeptide, and now we need to terminate because we have reached a stop codon or a termination codon. There are only a few of them. So, most of the time, these stop codons occur, in regions that are untranslated, so they don't actually make a protein. But in those long open reading frames, like we said, we've got long stretches of codons that are uninterrupted by stop codons. So when we finally get to a stop codon, that's going to tell the ribosome, okay, we're done. So that stop codon gets to the A site. And then there are termination factors or release factors, which will cause the ribosome to dissociate. So there's RF1, 2, and 3, and they function to hydrolyze the terminal peptidyl tRNA bond. So what that means is you need to hydrolyze it and get it off of the tRNA, right? We've got this long peptide chain on a tRNA that's stuck in the P site, and we need to get that off of the tRNA. So we hydrolyze that. We also have to release the polypeptide and the last uncharged tRNA. We also have to dissociate the whole ribosome, and again, we're talking about bacteria, 70S bacteria, and all of its subunits. Eukaryotes have one release factor, ERF. So this is one of those exceptions to the rule. Bacteria have 3 different release factors, and eukaryotes only have 1. There's always an exception. You just have to look. Sometimes you have to look a bit harder, but there's always an exception to the rule in biochemistry, which is one of the things that makes it really tough, but really interesting. So to carry on in bacteria, once we have released that protein, we also need to release the ribosome and recycle it. So the ribosome recycling factor, or RRF, uses GTP hydrolysis to help the ribosome dissociate. So it's going to, we've got the release factor binding to the A site here. We're going to hydrolyze that bond so that we can release a free polypeptide, and it's got a free carboxyl end. But we've still got this empty tRNA here that needs to move. So we're going to use EFG again, and we're also going to use RRF. And those two things together are going to do 2 things. Remove that empty tRNA and make it dissociate. And it's also going to remove that release factor that bound to the stop codon. So that translocation thing happens again to get rid of everything that's bound. And once all of those things are gone, they dissociate, then you've got the 30S subunit that has IF3 bound which helps promote the dissociation of the tRNA. Remember that IF3 can bind in the E site. And then it's associated with the 30S subunit, and it's ready for another round. So that 30S subunit can go on and make another initiation complex, and that 50S subunit can top off an initiation complex once all the other things have been recruited. So we recycle ribosomes. There are so many pieces, so many proteins and RNAs involved that you wouldn't want to have to remake the ribosome every time. So these ribosomes get recycled and reused a lot before they get degraded. So let's talk about the energy cost of protein synthesis and how it contributes to the fidelity of the process. So there's at least 4 high energy phosphate equivalents that are required to generate each peptide bond. Think about that for a second. If you've got even a 50 amino acid protein, 50 times 4 is 200 high energy bonds. So aminoacyl tRNA formation uses 2 high energy phosphate groups, because remember, we are adding on that AMP to the amino acid, and we're also hydrolyzing the pyrophosphate. So that's 2. We're also using a GTP to cleave during the first elongation step. We also cleave one during translocation. And then, if there's some kind of incorrect amino acid that's added, so it's the wrong tRNA, then you're going to consume energy for proofreading. So there's a high investment of energy to guarantee that you're making the correct protein. But when you think about all of these proteins, misfolded proteins or proteins that have inactive sites or too active or all these other things that can lead to disease, it makes sense that you don't want to have a protein that has a bunch of mutations in it. We can field a few mutations, and the genetic code helps to silence those mutations so that we don't see a real effect. But if protein synthesis did not have high fidelity, we would see a lot more issues with cell functionality. So let's talk about what it looks like in bacteria. We've been talking about DNA replication, RNA, making messenger RNA primarily is what we focus on, and then protein synthesis. Well, in bacteria, transcription and translation are very tightly coupled. So you have DNA and you've got transcription going on. So there's RNA polymerase, you're making a messenger RNA transcript. As soon as that transcript is peeking out of that RNA polymerase enough for a ribosome to sit down on it, it's going to. I've got music in my head now. If you get the reference, you're an old soul like me. Anywho, we have this ribosome that is now sitting on this newly forming messenger RNA. And as it chugs along, it's going to make a protein. And then once that starting AUG becomes available, then you can have another ribosome sit down and start making another protein. So we're making multiple copies. And then as that one chugs along, then you can have another initiation complex coming together around that starting AUG and start making another protein. So these two processes of transcription and translation in bacteria are very tightly coupled. And the complex of ribosomes and RNA polymerase all together is called the expressome. And you can actually see this in bacteria. So all of these little dots and clusters are all polysomes. They're clusters of ribosomes. and they are gathered around the DNA. So I think that's pretty cool. But the reason why this can happen in bacteria is because they don't have any compartments. Their DNA is just hanging out and just chilling. Eukaryotes, we don't do that. So we can't exactly couple those two processes. Now, stage 5, we're talking about eukaryotes only, primarily. Yes, there's some modification in bacteria, but eukaryotes are the champs when it comes to protein modification and folding and changing things. So newly synthesized polypeptide chains have to undergo folding and processing. Sometimes this can happen during synthesis, but oftentimes there are processes that happen after the polypeptide has been released from the ribosome, and it will assume its native conformation. We think about this happening, it just sampling, all of the conformations and just ending up on the correct fold. But in reality, we need chaperones and chaperonins, which we talked about a little bit when we were talking about protein structure way at the beginning of the course. So these proteins assist newly formed polypeptide chains to fold by restricting formation of aggregates and restricting certain conformations. So they physically limit the conformational space by simply binding to certain areas. So in bacteria, you have GRO-EL and GRO-ES, and in eukaryotes, you've got HSP60. And these processes of helping proteins fold requires ATP. So again, we've already made the polypeptide, but we're still investing energy to make sure not only do we have the right primary sequence, but that we've got the right secondary and tertiary structures. So on the left, for part A of this figure, we're looking at GRO-EL and GRO-ES. So this is bacterial. And what happens is you have essentially what looks like a cylinder that's made of two parts. You've got GRO-EL and then a cap that's GRO-ES. So GRO-EL is this kind of cylinder. It's a multi-subunit. It forms a kind of like a barrel. And then GRO-ES has this multi-subunit thing that makes a cap. And two of these GRO-EL, GRO-ES complexes come together to make one major cylinder of protein folding happen. And you've got one at a time, you've got a new polypeptide come on in. GRO-ES says, all right, you're going to stay in here now. And ATP is hydrolyzed to help the protein actually fold properly. And once that protein is folded properly, then you've got the release of that protein. So the cap has to come off, and you have native protein, or you can have some kind of a slow folding intermediate. But either way, you've made some major changes to that initial polypeptide, and you've given it the space to sample the conformational space in kind of an assisted environment. And you'll notice again, so if you kind of draw a dotted line, it's really two complete units that associate together, and they take turns having ATP and ADP bound. So we just process the top part that's kind of like the gray color. That one just released a native protein. And now we can have a new one come in, or we can have a folding intermediate somehow delivered by another heat shock protein, which heat shock proteins are just chaperone proteins. So you can have another protein bind in this bottom part, and then you hydrolyze ATP again, and then you release something else. So it's really an ongoing thing. What we have in eukaryotes is fairly similar. And you can see we've got the two channels, the two pockets, to make something happen, to fold those proteins. And by the way, that was like a slice. If you would slice that whole complex in half and look on the inside, that's what part B was. I didn't mention that. But you don't really need to know like the structures per se, but just understand the purpose of these chaperones and what they do. And the fact that it costs energy. Okay. So after you actually have the native conformation of the protein, there are lots of other post-translational modifications that occur. And this is really where we beat the pants off of bacteria. So bacteria can do a lot of really cool chemistry. But we do a lot of really cool chemistry to modify our proteins to give them additional functionality. They're pretty much almost every protein that we make requires further processing by post-translational modifications to actually achieve their for real final active conformation. So it's one thing to have the right shape, but it's another thing to have all the correct decorations and everything else that you need to do your function. So forming the final functional protein can include one or all of these things. You could use a protease to remove residues. You can remove that formal group in bacteria. You can remove the N-terminal methionine residue altogether. You can remove additional N-terminal residues. You can remove residues from the C-terminus. In eukaryotes, like around half of the proteins that you see in their final active forms, they don't even have that methionine. So you can also acylate the amino terminal residue. So you can add a little functional group to the front of that protein. You can also modify carboxy terminal residues, the C-terminal residues. Lots of things can happen. A lot of proteins also have what's called a signal sequence, and that's 15 to 30 residues at the N-terminal end of some proteins. And this plays a role in directing the protein to its ultimate place where it needs to function. And these sequences are eventually removed by specific peptidases once they're sorted. We can have modification of individual amino acid residues too. We talk a lot about phosphorylation, particularly in the last portion where we talked about all the metabolic pathways with glycolysis and the citric acid cycle and all these other things, and how phosphorylation and dephosphorylation can regulate a lot of these processes. Well, those are post-translational modifications. So serines, threonines, and even tyrosines can be enzymatically phosphorylated with ATP. We can also add extra carboxyl groups to glutamate residues. And one example of this would be in the prothrombin protein that binds calcium, and that's involved in clotting. So thrombin. Prothrombin is the kind of like the pre-protein. It's not fully formed yet, kind of like proinsulin. There are some things that need to happen to it, some proteolysis, etc., that make it, a ready hormone in the case of insulin. With thrombin, it's the same thing. There are lots of residues, amino acid residues, that can be methylated. So lysine is a big one, and we'll see methylated lysines a lot when it comes to gene expression regulation. But it can also occur in some muscle proteins and in cytochrome C. Calmodulin also has a trimethylysine. So these are just examples. You don't have to know, specifically how to draw a methylysine per se, but just understand some of the different variety that you have with post-translational modifications, and when given, some of the purposes. And I'll make those more apparent as we get to them. So the carboxyl groups of glutamate can undergo methylation, and that will remove their charge. You can also attach carbohydrate side chains, and this is a big one. Your cells are coated in proteins, and a lot of those proteins that are out there in the extracellular space have proteins or proteins. Proteins have proteins on them. Proteins can have carbohydrates on them, and those are called glycoproteins. These carbohydrates are attached covalently either during or after polypeptide synthesis, and they can be attached enzymatically to asparagine residues, which are called N-linked oligosaccharides, or they can be attached to serine or threonine residues that have the hydroxyl at the end, and those are O-linked oligosaccharides. You can also have the addition of isoprenyl groups. So farnesylation, geranylation are just two examples. And those actually get added to cysteine residues at the near the end of the C-terminus. This helps anchor proteins to the membrane. So it's really important. There are proteins like the RAS proteins and lamins, nuclear lamins, that must be localized to their specific membranes in order to be functional. The RAS proteins are part of a lot of different cell signaling pathways, and if they're not localized to the plasma membrane properly, then they can't participate. So lots of work has actually been put in to study this this modification and to study its effects of losing the modification and how that can actually help treat cancers. So this is just an example of the farnesylation of a cysteine residue. So it's got a cysteine that's hanging out near the C-terminus, and there are isoprenyl phosphates that exist that are intermediates in making cholesterol. And so some of those intermediates are siphoned off to modify proteins. And that thiol is going to attack the carbon here, and you're going to replace that phosphate with this protein. And that's catalyzed by farnesyltransferase. There are other transferases like geranyltransferase that transfer a geranylgeranyl pyrophosphate to a protein as well. There's also the addition of prosthetic groups. So we talked about biotin, we talked about heme. All of those things get added to the protein after it's been fully formed. So I feel like this one really kind of helps bring everything full circle, this chapter, because we talk about these proteins in their fully formed native state with everything bound, but we don't think about the fact that there's a lot of energy invested in making the protein and ensuring that the correct cofactors are bound, that the right modifications are made so that they can be localized to the right spot. Like, there's a lot that goes on to coordinate protein synthesis and protein function. Lots of proteins are synthesized as really big inactive precursors, and then they require proteolytic cleavage to achieve their active forms. So I gave the example of proinsulin already. There are some viral proteins that require this. And we also talked about the zymogens, chymotrypsinogen, trypsinogen. They have to be proteolyzed first before they become active. So we talked about that when we were talking about some of those metabolic pathways in the earlier chapters. Disulfide crosslinks are also very important. So some proteins will either form intrachain or interchain disulfide bridges between cysteine residues. And you'll see that commonly in exported eukaryotic proteins just because the extracellular environment is very oxidizing. And so having those disulfide crosslinks helps protect the native conformation of the protein and prevent it from becoming unfolded. There are lots of antibiotics and toxins that inhibit protein synthesis. And these can be used as tools in the lab to study protein synthesis, but they can also be quite harmful. So purimycin is an inhibitory antibiotic that binds to the A site and terminates polypeptide synthesis. It's made by a Streptomyces species, and its structure is similar to the 3' end of an amino acyl tRNA. So once it binds to the A site, then that whole peptide is transferred to it, and then it gets stuck. And you have termination of protein synthesis. Tetracycline is another compound that will inhibit bacterial protein synthesis. So lots of things are specific to bacterial or eukaryotic. I'll try to point them out as we go along. So purimycin is specific to bacteria as well. Oftentimes there are other species, so some bacteria will make compounds that kill off other bacteria that are their competition. Or in the case of purimycin, it's made by a mold that will kill other bacteria. So it's a crazy world out there with those single-celled organisms. So tetracyclines inhibit bacterial proteins by blocking the A site and preventing the binding of a new amino acyl tRNA. So if you can't bind any new tRNAs that are charged, then you're not going to be doing any more protein synthesis. You're going to stall the ribosome, and if it's stalled long enough, then you might have one of those transfer messenger RNA try to come along and, scoot out the the antibiotic, or you're just going to arrest that ribosome and it's just going to, get degraded. Chloramphenicol is another one that you see in the lab. So when we're talking about making these expression plasmids and cloning plasmids in Chapter 9 with all the DNA-based information technologies, now we're looking at some of the compounds that we have antibiotic resistance genes for in these plasmids. Tetracycline, so that TET-R, chloramphenicol is another one. So it's kind of coming full circle. There should be lots of aha moments, hopefully, or questions that will lead to aha moments. But chloramphenicol inhibits bacterial, mitochondrial, and chloroplast protein synthesis. So it's pretty nasty stuff. And it blocks peptidyl transfer. But it doesn't affect eukaryotic protein synthesis. And what we mean by that is that the ribosomes in the cytosol are not affected. Mitochondrial protein synthesis is very similar to bacterial protein synthesis. So it's kind of lumped in along with that. Cyclohexamides are actually specific for eukaryotes. So this is nasty stuff. You certainly don't want to get any of this stuff in your body. It is a poison. And it will block the peptidyl transferase of the ADS. So you cannot have that reaction where the RNA helps to position the tRNAs to form the new peptide bond. So it is quite nasty. There's also streptomycin, which causes misreading of the genetic code in bacteria if you have low concentrations, and it will inhibit initiation at higher concentrations. So you can actually use streptomycin in the lab as part of a purification process if you want to get rid of as much nucleic acids as you can from your protein prep. You can precipitate ribosomes using high concentrations of streptomycin. And all of the RNA and all of the proteins and stuff that are associated with those complexes will become insoluble. I'm sure that you've probably heard of diphtheria. It's something that we don't have to worry about because we live in a country where the vaccine is available, and it's something that's just a part of everyday life. During the first year of life, you're given many different vaccines, and one of which is DTaP, and it has a vaccine for diphtheria as part of it. But in other countries that do not have the resources, diphtheria is still a very, very, very real issue. And the diphtheria toxin catalyzes ADP ribosylation of diphthomide, which is just a modified histidine. And it modifies an elongation factor and inactivates it. So if you have an elongation factor that is no longer active, then it is going to greatly affect your ability to synthesize new proteins. So this is still something that plagues many different countries, but thankfully we do not have to worry about that because we have access to the vaccine. Ricin is a toxic protein in the castor bean that inactivates the 6DS subunit of eukaryotic ribosomes, and it will depurinate a specific A residue. And that's real bad news because those ribosomal RNAs are important for the actual catalysis and for positioning of the RNAs and all sorts of things. So this is really bad. And it's a poison. It is just a straight up poison. So now that we've talked about all that fun stuff, right, about poisons, let's go back to destroying proteins, right? We're just on a destructive kick now, I guess. So this is the final section of chapter 27, protein targeting and degradation. In addition to the energetic cost of charging the tRNAs, bringing together the ribosome, making the polypeptide, processing the polypeptide and making sure it folds properly, modifying it, which some of those enzymes require energy to make modifications of amino acids and things. There's also a cost to transporting proteins. So there's a thermodynamic cost of protein synthesis that we then magnify by all of the processes required to transport proteins to their correct locations. This occurs in eukaryotic cells. Bacteria don't really have this problem the same way. Those signal sequences that I was talking about earlier, the short sequence at the end terminus that says, hey, I'm a protein that belongs in this compartment, they're typically removed during transport or right after they reach their final destination. And the end terminus for proteins that are slated for transport into the mitochondria, chloroplasts, or the ER. So all of these proteins have some kind of a signal sequence that tells all the cells what to do, where to put it. So post-translational modifications of lots of eukaryotic proteins begins at the ER, the endoplasmic reticulum. And these signal sequences mark most of the lysosomal, membrane, or secreted proteins for translocation to the ER lumen. So that's the inside of the ER. What we're looking at here is we have some viruses. We've got pre-pro-insulin, bovine growth hormone. All of these things actually have signal sequences, and there are cleavage sites so that after they get to their destination or along the journey, they get cleaved off. Signal sequences vary in length, but they're still on the relatively short side. They have usually a good stretch of hydrophobic amino acid residues, and they have a positively charged residue that's usually near the end terminus. They have a short polar sequence at the carboxy terminus, meaning the part that's closest to the start of the actual protein. So if you have your signal sequence, abbreviate that SS, and then you have the rest of your protein, you'll have some kind of positively charged residue. So you might have a few of those at the end terminus. And then you'll have some hydrophobic ones. So that H is for hydrophobic, not histidine, hydrophobic. And then you'll have a short polar sequence. that's near the actual start of the protein for real, so at the end of the signal sequence. And that's the general architecture of a signal sequence, but this is still pretty loose, right? The signal sequence is always synthesized first. It's at the end terminus of the protein, so it's the first thing that happens. And there's a particle it's a complex called the signal recognition particle that recognizes and binds to the signal sequence right at the ribosome. It binds GTP, and it will halt elongation at around 70 amino acids. So you have that signal sequence fully out, and then you have a few more residues after that. And then the SRP says, all right, hold up for a second. And it will redirect the ribosome and the mRNA and the incomplete peptide, polypeptide, to a receptor, the SRP receptor that's on the cytosolic face of the ER. That receptor is also bound to GTP. This is called the peptide translocation complex, which interacts with the ribosome and accepts that newly forming polypeptide. SRP is going to hydrolyze its GTP, and in combination it will also dissociate from the ribosome. So all it does is deliver the whole kit and caboodle to the ER so that it can release that polypeptide into the ER lumen. The elongation of the polypeptide continues, and that growing peptide is fed into the ER lumen through that translocation complex. The signal sequence is removed by a signal peptidase within the ER lumen, and then the ribosome dissociates from the ER and it's recycled. So this is just a pictorial representation of what we just talked about. The first thing that happens is you make your polypeptide. You eventually have that free signal sequence out there, which will be bound by SRP. Once you get a little bit more of that polypeptide made, eventually you're going to relocate that ribosome and the mRNA to that SRP receptor, which is coupled to a peptide translocation complex that's on the cytosolic face of the ER. Once that happens, the ribosome is going to continue to make that polypeptide, and SRP is going to hydrolyze its GTP, and then it's going to leave. Once the ribosome, which is a ribozyme, but once the ribosome finishes making that polypeptide, and that polypeptide exists inside of the ER lumen, that ribosome is going to be dissociated just like it normally would if it was just hanging out in the cytosol, and it's going to go off and be recycled to do the whole shebang all over again. So glycosylation plays a really big role in protein targeting. It's like another language. Just like the amino acids, glycosylation can tell you, those different patterns can tell you where a protein belongs. And N-glycosylation occurs in the ER lumen. It involves a 14 residue core oligosaccharide that's built up stepwise. Most of the O-glycosylation actually occurs in the Golgi complex or in the cytosol. So this is just a demonstration of how these core oligosaccharides are built that end up being part of the glycans that go onto the glycoproteins. And you're not responsible for knowing all of this, but just understand in general what happens. You build up these core oligosaccharides by having a protein that is on the, it's like in the ER membrane. And you build stepwise these different sugars into these complex formations. Eventually, there's a translocation that happens where all of this sugar craziness gets flipped to the inside of the ER, and it's now in the ER lumen. And once it's there, it's available for a new protein or polypeptide that's being formed to attack and say, hey, you know what? I want that decoration. That's going to look good on me. And so you can have N-glycosylation within the lumen as the polypeptide is being made. It could also happen again after it's been formed, and then you can still have glycosylation. Tunicamycin blocks the first step in the synthesis of that core oligosaccharide. It mimics the structure of UDP-N acetylglucosamine, which is key in creating these monstrous, complicated sugar structures. So once we have a protein made and it's maybe partially decorated in the ER, the proteins are going to travel from the ER to the cis side of the Golgi complex in transport vesicles. So to remind you of your, what a cell looks like, you've got the nucleus with the nuclear envelope, which is, it's got some pores in it. And we'll talk about those because we're going to talk about them a little bit later. And then you've got around that the ER. And the ER has the rough ER, which has the ribosome studded, what we were just talking about, where they're spitting out those polypeptides into the ER lumen. And then there's the smooth ER, which is used to create transport vesicles. And those vesicles end up merging with the cis side of the Golgi complex. So the Golgi complex is like the mail sorting center. So let's say that, the ribosome is where you put your stuff into the mailbox, and then a mail carrier will come and pick it up and take it to the nearest post office. So the mail person taking it from the, would be kind of the vesicles from the smooth ER to the cis side of the Golgi. Once we're at the mailing center, the Golgi is going to sort things and send it out where it's supposed to go. And so it goes through this process. And there's some modifications that can happen in the Golgi too for proteins while they're being sorted. And then on the trans side, you've got more vesicles being formed, and those proteins are sent to where they're supposed to go, the lysosome, or they're getting transported out to, be in the cytosol, or they're going to go to the plasma membrane where those proteins are then secreted. So the Golgi tells everything where it's supposed to go. The phosphorylation of mannose residues on lysosome targeted enzymes is really key. So this is one of the pathways of how the Golgi kind of sorts and sends things off that's kind of understood. The truth is, how the Golgi sorts and gets everything right is still largely a mystery. It's a beautifully complex system that is quite difficult to understand. But N-acetylglucosamine phosphotransferase recognizes an unknown structural feature of hydrolases that are destined for the lysosome. We're not going to stay too heavy on the, understanding the Golgi complex and how it sorts, because we just don't know. But there are some things that we do sort of understand. So what I want you to get from this part is, in general, what does the Golgi do? And understand that there's still lots of question marks here. So we talked about sending things to all over the place, but we didn't talk about sending things to the nucleus. And that happens quite often. The signal sequences for nuclear transport are actually not cleaved. So those are called nuclear localization sequences, or NLSs. And they provide a signal sequence that will target a protein to the nucleus. It's not cleaved, because it allows for these proteins to be, imported and exported without having to have another signal tacked onto it. Nuclear importation is mediated by several proteins. We've got importans, alpha and beta. And then we've also got GTPases. There's a GTPase called RAN, which is it's a cousin of the RAS protein, so it's related to RAS. So this is what happens. I'm going to tell you first what's in B. What we're looking at is the nuclear pore complexes. So all these kind of big raised spots, these are pores that allow proteins and things in and out of the nucleus. And of course, it's regulated. So that nuclear envelope that surrounds the nucleus is not 100% sealed off because there needs to be exchange of proteins and other goods across the lines. So what happens is you have a nuclear protein that's been made that has an NLS, that nuclear localization sequence. It's going to be bound by importance alpha and beta. They're hanging out in the cytosol. Once they're bound, this gives that new protein that needs to get to the nucleus access. It's going to travel through that nuclear pore complex where it will encounter RAN that's bound to GTP. Just like RAS, RAN binds GTP and it hydrolyzes it at a particular rate that will regulate some process. And in this case, the process is regulating importation of nuclear proteins. So RAN GTP will come in and it will bind to the beta, the important beta. There's a Cas protein that will bind to the alpha, the important alpha, which will then bind RAN GTP. This will enable the importans to be exported from the nucleus. And in the process, the RAN GTP will hydrolyze its GTP to GDP. Now we've got RAN GDP in the cytosol. In order for it to act as a transport, it needs to have GTP bound. So as part of this cycle, there's NTF2, which will bind to RAN GDP and bring it back into the nucleus, where it will encounter its GEF, guanine exchange factor, or guanine nucleotide exchange factor. And that just is a protein that will bind to RAN GDP and facilitate the swapping of GDP for GTP. It enhances the rate that it will exchange that nucleotide. Now that we've got GTP bound to RAN, that NTF2 that escorted it across the nuclear envelope and through the pore is now dissociated, and that RAN GTP can then go on and bind more importans in the nucleoplasm. So it's like two processes that are running simultaneously, and one is supported by the other. So that's definitely a process you'll want to understand. And if there are issues with this process, let's say there's a mutation or something that causes RAN to constantly bind GTP or never bind GTP and it's always in the GDP bound form, what might happen? Think about it. So bacteria also use signal sequences for protein targeting, but it's just that eukaryotes they do it to the most extra level possible. So they use N-terminal sequences to target proteins to their inner membrane versus the outer membrane, or the periplasmic space that exists between them. And if you're saying, doesn't that sound a lot like what the mitochondria has? Yes, it does. There's also exporting proteins to the extracellular medium. So that's how toxins get out, right? You label a protein that a bacteria has made for secreting it out, not just into the periplasm, but all the way out into the outside world, the extracellular medium. So this is a model for protein export in bacteria. We've got the sec protein, sec B and sec A. Sec B is a chaperone protein, and it will bind a protein that needs to be exported. It will take that protein and escort it to sec A, which is a receptor, and it's also a translocating ATPase. So it hydrolyzes ATP, and in doing so, it allows the translocation of a protein from the cytosol to the periplasma. There's a complex, sec Y, E, and G, that makes up the translocation complex that sec A is associated with. So sec A is associated with the membrane on the cytosolic side of the inner membrane. Then you've got sec Y, E, and G that are in that inner membrane. And upon ATP hydrolysis, you're going to allow that protein through the translocation complex to enter the periplasmic space. Now, sec B doesn't stay around the whole time. When it brings that protein to sec A, it's going to be replaced or kicked out when ATP binds. So ATP binds and sec B leaves, and then you have hydrolysis, which leads to the translocation of the protein. Cells import proteins and they export proteins. And this happens, one of the ways, by receptor-mediated endocytosis. So there's different ways that you can do this. There's clathrin-dependent endocytosis, caveolin-dependent endocytosis, and there are pathways that are independent of clathrin and caveolin. With clathrin, there's a protein called dynamin along with clathrin. And clathrin just kind of makes a structure that almost looks like a buckyball. And then you kind of pinch off the membrane to keep in whatever it is that you want in that vesicle. So that's how you can import proteins or receptors to bring them inside to be recycled or to, deliver a signal to the inside of the cell. It's a similar thing with caveolin-dependent endocytosis, where you have caveolin all along the outside, and you have the pinching off of, a little bleb of membrane by dynamin, and then that will go and bind to a caveosome to release its contents, or it will go into the formation of an early endosome, which all of these things can do. So clathrin-dependent endocytosis, we create these coated pits. And these are the places where the membranes have a concentration of endocytic receptors. So things that are related to, we should form, a little pouch and close it off. So all the receptors involved in that. So they're coated on their cytosolic side, so on the inside of the cell, with clathrin. And clathrin is literally forms a cage like structure. And then dynamin is a GTPase that will pinch off the endocytic vesicle. So like I said, it's just kind of like you pinch it off. Like if you've ever made dough and you're trying to pinch off a little piece, you literally kind of squeeze it with your forefinger and your thumb, and you can squeeze off a little piece or a little dough ball. It's the same idea, only with membrane. Protein degradation. We're finally getting to that now. We talked about all the different ways that we can ship proteins all around the world, aka the cell. We could ship them out. We could bring them in. Now we have to talk about how we degrade proteins, because that's a major part in controlling all the different functions that go on in a cell. So there's specialized systems in all cells that mediate protein degradation, and it's critical to cellular proteostasis. The half-lives of eukaryotic proteins wildly varies, from 30 seconds to many, many days. Defective proteins, short-lived. Don't got time for it. Enzymes at key regulation points in metabolic pathways are also short-lived, so that you have very quick responses to turning on and turning off a particular signal. You don't want to keep a pathway going when you don't need it, and you don't want to turn it off when you really mean to turn it on, right? So we have to have ways of very quickly turning on and off these pathways. Hemoglobin is an example of a protein that is very long-lived. It usually will live the lifespan of the erythrocyte that it's hanging out in. So that's like 100 days, somewhere in that ballpark. Protein degradation in eukaryotes, one of the things that we use is ubiquitin, the ubiquitin ligase system. This is something that you may not have heard of in biology. It's relatively new. I say relatively, but it's not something that was known for 100 years. So that's what I mean by relatively new. Ubiquitin is actually a small protein, and it is covalently linked to proteins that are slated for destruction, and it requires ATP. This is a highly conserved protein, so it looks the same across the board pretty much in eukaryotes. The ATP-dependent pathway includes 3 different enzyme types. There's the E1 activating enzymes, E2 conjugating enzymes, and E3 ligases. So each one of these enzyme types, there's different flavors so that you can bag and tag different proteins that need to be degraded. So the first thing that has to happen is you have to attach ubiquitin to a target protein. And there's still a lot of research going on to understand what it is exactly about a protein that labels it, that the system says, hey, we need to tag this thing for degradation. So you've got ubiquitin that's got a free carboxyl N. You have an E1 and ATP. That E1 has a free thiol. You're going to use that ATP to activate the ubiquitin and get it loaded onto the E1. Once you have E1 loaded with ubiquitin, it hands it off to E2. Once the E2 is loaded with the ubiquitin, again on another cysteine residue, the E2 is going to arrive at the E3, which is like a scaffolding protein, and it's got places for the E2 to bind along with the target protein that needs to be ubiquitinated. That reaction happens, and then you have a ubiquitin added to this target protein. Now, one ubiquitin usually isn't enough. You actually need lots of ubiquitin molecules, and sometimes even on several different residues. But these ubiquitin proteins are added onto lysine residues of the target proteins. So you may have lots of ubiquitins on several different lysine residues within that target protein before it gets degraded. Once a protein is tagged for degradation, it is going to ultimately end up in the proteasome. We've got lots of zomes, I realize. Sometimes it's hard to keep them straight. The 26S proteasome is like a barrel type structure, so it's sort of reminiscent of the GROES, GROEL type structure where you have the barrel and the cap and you allow proteins to fold. But in this case, you're inviting the proteins in. You're saying, hey, come on in. The water's fine. Warm place, cozy. And then you chew them up with proteases. It's pretty evil, but you have to degrade proteins. It's just a part of life. So the 26S proteasome is a very large complex that degrades ubiquitinated proteins. It's pretty highly conserved. So you've got this 19S regulatory particle on the top end and on the bottom end. And this 19S regulatory particle will recognize the polyubiquitin on all the different lysine residues. The substrate protein will interact with the proteasome, and it'll say, oh, hey, yeah, I'll check it out. And then we've got this 20S core, which is made of multiple alpha subunits and beta subunits. The beta subunits are where the action is. Those have the different proteases that will chew up the protein and yield, different size peptides. So the identity of the first residue has a profound influence on the half-life of a protein. And for whatever reason, N-terminal residues that are alanine, glycine, methionine, you can read, those proteins have a fairly long half-life, more than 20 hours. But some of these other residues at the N-terminus have very short half-lives. Why? I don't know, but they do. It's an observation, but we still don't understand why. What is it about having these different residues at the very beginning of the protein that marks them as stable or unstable? Who knows? Maybe you'll figure it out. Anyway, that's it for protein metabolism. It was a long ride, but we finally got there. We talked about how we make the protein, how we modify it, ship it off to where it needs to go to do its business, and once that protein has to retire, hang up the cape. I don't even know what that really means, but you know what I mean. You're retiring, right? I'm thinking like a boxer, who goes out into the ring and has like a cape or something on, and they finally hang it up because they're retiring. Just walk with me for a second. The protein is retiring, and now we have to take it to the proteasome for the final resting place. So we just walked through all of that. And what I want you to do is to think about the ways in which DNA, RNA, and protein metabolism are similar. Think about the similarities and differences between eukaryotes and bacteria. And think about all of the reasons for why it's important for energy to be used in these processes and why it's so important to have high fidelity. So those are just some of the reasons, some of the ways that you should be thinking about reasoning through all the information that you've just gotten. So hopefully there's been some aha moments in bringing together some elements from the beginning of class to now, and we'll do some more work on this in class. Thanks for watching as always, and be safe.